researchers have uncovered the role the thymus gland plays in creating and training specialized anti-cancer white blood cells, known as gamma delta T cells. We have large numbers of these specialized cells in our blood and tissues, which accumulate as we become adults. Until this study was published, it was unclear how these cells develop in our body. Many experts assumed that after birth, the thymus played little role in the development of these cells as we age, but we now know this little unsung organ helps the body prepare for a lifetime of good health. These gamma delta T cells have been implicated in a variety of immune settings, including antimicrobial and anti-cancer responses. For example, antigen-driven expansion of these cells in the lung improves host immunity toward tuberculosis and reduces disease burden in macaques. These cells can also kill transformed host cells that upregulate a self-phosphoantigen called isopentenyl pyrophosphate, IPP, suggesting an important role in anti-cancer immunity. In promising trials, these cells have been targeted in cancer treatments, including treatment of bone and colorectal cancers. Consequently, understanding the factors that govern the frequency and development of these cells could lead to strategies to manipulate these cells to treat human diseases.
A third new Alzheimer's drug is expected to be approved by the Food and Drug Administration, FDA. The field is beginning to show progress in the fight to slow the disease. But the drugs work best for those in the earliest stages of Alzheimer's, and other therapies will be needed to help those with advanced disease. Donanemab is a monoclonal antibody, like the two earlier Alzheimer's drugs, aducanumab or Aduhelm, and lecanemab or Leqembi. These drugs attack plaques in the brain that are made of a protein called amyloid. They disrupt cell function and lead to the rapid spread of another protein called tau. Both amyloid and tau contribute to the development of Alzheimer's disease. The trial showed donanemab slowed cognitive decline by 35% compared with placebo in patients with low to intermediate levels of tau in the brain. These results are similar to those reported with Leqembi, which received FDA approval earlier this month. In the donanemab trial, patients also experienced a 40% lower risk of progressing from mild cognitive impairment to mild dementia, or from mild to moderate dementia. Donanemab was better at removing amyloid plaques compared to Aduhelm and Leqembi. It reduces tau concentrations in the blood, but not in a key area of the brain. While these results are encouraging, but still an in-depth analysis still is needed to understand how these findings affect patient outcomes.
This might be a surprise, but mosquitoes turn out to be the world's deadliest animal. They carry malaria, which kills hundreds of thousands of people each year. New research tries to make these pests into allies in the fight against the disease.
Malaria parasite doesn't make mosquitoes sick, so mosquito immune systems don't fight the parasite. But here's a fun fact. There's a little rodent that can fight off malaria, the mouse. The team used a gene editing technique called CRISPR to put the mouse genes in mosquitoes. The results were published recently in the Proceedings of the National Academy of Sciences.
They work very well. They reduce the number of parasites in the mosquito, importantly in the salivary glands, which is where they would be before they're transmitted to a new human host.
This technique also allows the researchers to make the gene spread quickly. That means rather than having to release swarms of gene-edited mosquitoes, they could put out a smaller number. The engineered mosquitoes mate, pass on their genetic code, and that code rapidly fans out across the wild population. But genetically altering wild animals does not sit well with environmentalists.
There's no need to engineer a mosquito. There are naturally occurring methods to reduce malaria transmission that are already proving successful around the world. There's a new malaria vaccine that's being piloted. Why take unnecessary risks and release a manipulated species that can't be recalled once it's released into the wild?
Anthony James believes the risks would be very low. His lab is now working on developing the mosquitoes for a field trial, which he hopes could be conducted on an island or in another isolated location.